Okay. Hello, everybody. I'm Dan Brown. I'm here to welcome you to the first of our hybrid virtual in-person SEFS seminars in our fall 2021 series. Welcome, everybody. We are looking to engage everybody in the room and on Zoom, and we're going to use the chat and or a Q&A function. If you have questions throughout the talk, feel free to use them. We'll read them to the presenter at the end during the Q&A session. I'm very pleased to welcome today David Smith, who is a professor in the Department of Health Metrics Sciences and the Institute of Health Metrics and Evaluation in the School of Medicine at the University of Washington. Before this appointment at the University of Washington, he was on faculty at Johns Hopkins University, University of Florida, a mathematical epidemiologist at the Fogarty International Center, the National Institutes of Health, and before that in the Department of Epidemiology at the University of Maryland School of Medicine. Professor Smith heads the Malaria Control and Elimination team at IHME. His research focuses on infectious disease dynamics as a key component of health analytics for robust policy and adaptive management, especially robust analytics for malaria policy. He defines robust analytics as the quantitative synthesis of evidence to create maps and models for policy analysis that fully propagates uncertainty. The team develops theory, methodology, and software for landscape analysis and simulation in collaboration with partners at the local burden of disease, the global burden of disease projects at IHME, at the Clinton Health Access Initiative, and at the Malaria Atlas Project, among others. He has a PhD in ecology and evolutionary biology in Princeton University, from Princeton University. Please join me in welcoming Dr. Smith for today's presentation.
Thank you, Tim. So I am going to give a talk where I can't see any of you. Before I start, I just want to express the hope that someday we can meet face to face and we'll be back to normal. Let's see, can I go? Okay. So I've been working on malaria for about the past 23 years, on and off. I've worked on other things, but mostly malaria, and I've really enjoyed doing it. Today, I'm going to talk about a bunch of research that I've been doing over the past 20 years to get to a point where we could actually give reasonable answers in a reasonable amount of time to hard policy questions. So I'm going to assume that most of you aren't malaria experts, although I can see a couple in the room here. So I'm going to start very basic, and I'm going to try not to be too technical in my remarks today. And I hope if you have questions about any of the technical things, they're in the papers, and I'd be very happy to discuss them with you. So the first thing is to say human malaria is a disease. That is to say, it's something that makes us sick and kills us. Even today, it's responsible for somewhere around 400,000 deaths a year. In addition to deaths, there are about maybe 10 times that many cases of severe malaria with chronic sequelae of various sorts that leave people disabled for a lifetime. There's also a heavy burden of uncomplicated malaria accompanied by fever. In a recent study, we attributed on the order of 80 days of fever per year. It's a self-reported fever where you ask somebody how bad do you feel sick, and they say yes. 80 in kids, about 55 in older kids, and about 45 in adults. It's also an infection of red blood cells, so it contributes to anemia and the burden of severe anemia. And it's also a cause of severe malaria in pregnancy, which leads to bad outcomes, including low birth weights and all of the health effects that follow from low birth weights. It's also been one of the main, for those of you who are biologists, You'll remember GBS Haldane's hypothesis about sickle cell anemia. It's been one of the main reasons why we have these genetic blood disorders that are in the haploid state protective against malaria. So this includes sickle cell anemia and thalassemia, among others. I'm gonna have to move this just a bit if I can, just so you can see the rest of the screen. Part of what makes malaria such a bad actor is that malaria epidemiology is really complicated by the fact that immunity to malaria is never completely protective. And because of this partial protection, a person can get malaria over and over again. And repeated bouts of malaria have long-term health consequences. One of the consequences is reinfection is common. You get it over and over again in multiple bouts of disease. You can also at one point in time be carrying multiple parasites. And if you think about it, this is a sexually reproducing organism. Sexual reproduction takes place in the mosquito. So to have outcrossing, you have to take up two parasites from different infections in the same mosquito. And outcrossing is something we measure at pretty high rates in parasites. So outcrossing is a common feature of these systems. So superinfection, or having a high multiplicity of infection, is also common. Malaria can be treated and cured, and in fact, if you treat it soon enough, it prevents a lot of severe malaria. But of course, we're in situations where access to healthcare is pretty poor. And so this is one of the reasons why the burden remains so high. It's also worth pointing out that health outcomes can vary with genetics, nutrition, and co-infection and other things. So human malaria is a disease. Human malaria is a disease caused by infection with a parasite. I would be remiss if I didn't spend at least some time talking about the parasite life cycle. Seems to be a requirement of most talks. The main features of this life cycle is that it takes two infectious bites by mosquitoes, or two bites by mosquitoes, for a parasite to complete its life cycle. One to infect the mosquito, at which point parasite undergoes, invades the gut, undergoes replication, flows through the hemocoel up to the salivary glands where it's ready to be there in the next bite. Goes through the skin. The second bite introduces sporozoites into the blood, which go into the liver. After 6 days, they come out of the liver and start replicating in blood. That replication cycle is what makes you sick, what causes most of the disease. And then at some point, these differentiated into a male and female called a micro and macro gametocyte to be transmitted by the mosquito. Excuse me, that was long. You've all now been inducted into the parasitology world. So there are about, depending on how you count it, most of you probably learned 4 species of parasites that cause malaria. That picture has changed a bit. Ovale turns out to be 2 species, not 1. Sympatric species that don't mate. And we have 2 new zoonotic species, one widely recognized nullside, the other one apparently also in an enzootic cycle called P. simium. Plasmodium falciparum is the one I'm going to talk about today. It's the one that has the biggest burden associated with it, and to a first approximation, almost all of that burden is in sub-Saharan Africa, for complex reasons related mostly, I think, to the vectors, but probably also to public health. Malaria is a disease caused by infection with a parasite that's transmitted by mosquitoes, and the mosquitoes are every bit as complicated as the parasites. So they have they're holometabolous insects. That means that they have a life cycle which includes eggs, 4 larval instars, pupae, and adults. All of the aquatic stages, or all of the immature phases, live in water. And so when the adult emerges from the water and flies about, it lays eggs in lots of places. So this life cycle is common among insects. There are about 70 species of mosquitoes, all in the genus Anopheles, that transmit human malaria. Once the adult is emerged, it does something that permits transmission. That is, it feeds on human blood. Actually, mosquitoes feed on vertebrate blood, but it's feeding on human blood that actually permits the parasite to complete its life cycle. And that blood mainly gets turned into eggs. But mosquitoes do other things. They mate, they sugar feed, they rest because they don't like to fly around heavy. And so there are lots of things that, activities that have to take place for mosquito to complete its life cycle. In the process of doing that, they move around a lot because they have to go from one resource to another. And during this process, they're also moving the parasites around. And so understanding the infection dynamics is pretty critical. I should say, because I'm sure it'll come up in the questions, the mosquitoes are cold-blooded and they're affected by weather a lot, including wind, rainfall, and temperature. And in particular, rainfall ends up playing a dominant role in this because it creates habitat for the mosquitoes to breed. The other thing about malaria that's really important is that we have a lot of tools out there to control it. I've been inundated with phone calls today and questions about the new announcement about the approval for the first time in the history of the world of a malaria vaccine. So it's now available. We can have a longer chat about this. I hadn't planned on talking about it, but since you've seen the headlines, if you want to ask later, let's talk about it later. The main tool has been to cure disease has been anti-malarial drugs, starting from a very long time ago when we could cure malaria with cinchona bark. Nowadays, the first-line therapies are artemisinin combination therapies, and they're highly effective, but we are seeing some resistance. Delivery turns out to be a critical component of this. How do you get the drugs to people? If you just wait for them to come to you, that's called ordinary case management. But there are other ways of delivering it. Most of us, if we went to Africa, we would chemoprophylax. We'd take drugs to protect ourselves from ever being infected. But in Africa, that's not common unless you're very wealthy, in which case we have lots of other ways. Visiting people frequently and asking them to test themselves frequently and doing all kinds of things to try and give them better access. The other side of things is vector control, where we try to do things to suppress transmission by mosquitoes, including suppressing mosquito populations themselves. So I will talk a little bit about this later, but the predominant tool lately has been insecticide-treated bed nets, but it's one of many possibilities, including sort of more traditional larval source or environmental management tools to get rid of aquatic habitats or treat them so that they don't produce. So this is complicated already. I've spent a long time just giving you background. One of the ways to understand it is by using mathematical models. And there was a very simple model that was developed more than 110 years ago now by Ronald Ross. It was formulated as a set of ordinary differential equations. I've showed you up here on the left side of the screen at the top, you see the equations clipped from a 1911 Nature paper. So this was, what the model looked like then. And it was analyzed by Alfred Lotka. So most of you will have know of Lotka from other things like competition in ecology or demography. In demography, he invented a term called R0, which was the number of female humans born to a female human. And George MacDonald borrowed that idea for malaria. And he invented a formula that took parameter names that had been assigned to the traits that entomologists had been measuring. And he emphasized the importance of, what was the importance of those in the ability of a malaria parasite to spread from one host to another. So this was a measure not of reproductive success by female humans, but by parasites. And the number of offspring is measured in the number of new humans infected per human, or no mosquitoes infected per mosquito. So this is just a sort of, bitten by mosquitoes, survive, continues to live long enough to give future bites, gets more bites. Some of those cause an infection, and that infection lasts for a number of days. That was the logic that went into that bite, and I'm going to come back to it. On the right-hand side, you're seeing a prettier version that I developed as sort of an essay overviewing malaria and other mosquito-borne diseases with the same parameter names and sort of developed with an artist, so it looks prettier. But it's essentially expressing the same process. Now, an important idea, and this was sort of recognized by Ross himself and others, is that this was a very simple description of malaria. If we want to get into malaria and actually make it work, we have to struggle with this big question of how do we represent all of the complicated things happening in mosquito, human, and parasite populations for transmission to occur. We're getting into a question that is actually discussed more in the arts and humanities than it is in the sciences, but I'm going to raise it today as a scientist to say we have an issue here about how much we're going to be mimetic. How much are we going to try to imitate the systems that we're looking at? And just for a little, artistic flair, I put up a postcard from Picasso on the right. At least I bought it as a postcard. It's very pretty. But representation is something we do. And in fact, models are something we all use in our everyday life. And it's sort of hard to think of doing science without having some sort of abstract representation of the world. One of the reasons this becomes so critical is that because of the complicated nature of transmission with mosquitoes and humans, that there's a great variety to the way malaria gets transmitted as you move from place to place. Different species of mosquitoes, different ways that humans and mosquitoes meet up. It was encapsulated in this quote by Hackett in this paper from 1937 that I'm going to read because I think it's a lovely quote. Everything about malaria is so molded and altered by local conditions that it becomes 1,000 different pieces and epidemiological puzzles. Like chess, it is played with a few pieces, but it is capable of an infinite variety of situations. What I'm showing you on the right is, in picture form, one of those situations. So this is Kilifi on the west coast of Kenya, just up from Mombasa. If you guys have been there, there's a Wellcome Trust Center. And this is from the DSS. They've been recording all of the hospital visits. And over a 10-year period, this is the average fraction of times that a person from that whole household showed up at the hospital infected with malaria, the malaria positive fraction. And what you're seeing here is an enormous amount of variability in the incidence of malaria over very short distances. And it's kilometers, I think, at the top that you can see. So you can see patches of blue, patches of red, and then blue and red almost right next to each other with enormous differences in the risk. All right, so just to say that as of 1910, we wanted to sort of see what people had been thinking about malaria in mathematical models, so we conducted a systematic review. And this is our pictorial representation of the things that had been included in those models. So we, again, with the help of an artist, tried to sort of represent a very large set of considerations. And we learned a few things from this. One of them is it's a very bad idea to do systematic reviews, which I think many of you have learned through hard experience. And the second one is that when people build models, they often start with the Ross McDonald model and they add just a few things. The biggest number was 1, followed by 2. Very, very few things had departed from the Ross McDonald model by more than 2 things, unless they had gone all the way to build a complex individual-based simulation model. So they'd gone all the way to representing all of the complexity all at once in these simulation models. So that was what we learned from this. And we sort of, what the problem with that is, I mean, it's great, you can deal with everything in these individual-based models, is they're stochastic and they're difficult to deal with. So we wanted to sort of do it a slightly different way so that we could, touch anything, we could translate evidence into policy. And so there are several steps here. One is you have to gather the evidence and then build a lot of models, including statistical and sort of simulation-based models, analyze them, and translate that analysis into advice. So that's why we've been developing something we call RAMP, Robust Analytics for Malaria Policy. So when I talk about RAMP, I mean, let me explain the title so you know exactly what I mean. When I say malaria policy, I'm not talking about generically. We are actually involved in discussions with, on the left-hand side, the Bioko Island Malaria Elimination Program. So this is where Mike Reddy did some of his thesis work. So Bioko is off the coast of West Africa, closer to Nigeria than to Equatorial Guinea, to which it belongs. But because of oil, it's actually, there's a lot of money there nowadays, and they're trying to control malaria. On the right-hand side, we have a collaboration with Jimmy Opaigo and the Uganda National Malaria Control Program in the Ministry of Health to help them sort of turn evidence into policy using some of these same tools. So when we talk about robust analytics, what I actually mean is robust is the opposite of frail. You want to give advice that's not going to fall apart when you start putting it under scrutiny. That's the basic notion. When I first started to do this, what immediately felt different or foreign was that this notion of robustness seemed to cut against the other notion that I'd learned about parsimony, to make a model that, as Einstein said, was as simple as possible, but no simpler. Robust basically means you want to ask this other question: would the analysis have come to the same conclusion if I'd formulated the model in a slightly different way? And there are a lot of decisions you make along the line in doing an analysis that can influence the outcome. So what you want to know is, would any of those decisions have really changed the outcome? Which seems to cut against this notion of picking the one simplest model to do your analysis and your inference with, or to finding the best model. So one of the ways to say it is, to think about all models as approximation. And Box is often quoted as saying all models are wrong, but some of them useful. But the really important question is, how wrong can a model be and still be useful? How bad can the approximation be? And that's the question we're going to try to get at here. So the other side of this is to think about the same kinds of questions, but flipping the script a bit. Are we dealing with a question about what we know, which would put it in the realm of science, or about what we should do, which puts it in the realm of policy and analytics? And the problem with what we should do is that something's going to be done regardless. So you want to do the best thing given the best available evidence, and you're going to update this over and over again. So you're going to do the same kinds of analysis in the same ways, but you're going to weigh the uncertainty very differently. In science, we end up being epistemologically conservative. We don't want to conclude something that's wrong, so we're very slow to believe something. Whereas in policy, we always have to act. In a nutshell, we want to be able to give advice that wouldn't change regardless of uncertainty. So the advice has to come in a certain way. We think this is what you should do, but we recognize, this is what we think will happen, but something else might happen because we're not sure about this, in which case you should measure something else. So it's always going to come with prescription about what to measure so you can figure out the next time around how to fix it. So when we talk about this, really, we're dealing with this problem of uncertainty. And this comes from a lot of sources, some of which we can fix and some we can't. One is that there are big data gaps. Just data hasn't been collected everywhere. And there are gaps in our uncertainty or in our knowledge that come from the fact that malaria really is variable as you move from place. There are nonlinearities. It's heterogeneous in a place, and there are therefore lots of identifiability problems, part of them coming from our inability to measure malaria as accurately as we'd like. So I want to talk about, the wrench analogy. So I want to sort of talk about the way we think about uncertainty by analogy to a crescent wrench. So I didn't realize Crescent was a brand, but adjustable wrench. So process uncertainty is, we want to build a model that will conform to the situations we find by twisting a few knobs. That's the wrench. Contextual uncertainty is all the information that tell us how do we adjust the knobs here. The third kind of uncertainty is much harder to illustrate with a crescent wrench. It's the fact that we don't actually know what the future looks like. So when we adjust to the situations that have characterized the past, as we project those into the future, we may get different situations. Evolution of drug and insecticide resistance are two, changing funding scenario is a third, changing weather or climate is a fourth that we can all think about. And those are very concrete ones that we worry about. So I want to talk about some of the attempts we've made. So the first thing is gathering spatial information. So remember, mosquito populations are driving this. Mosquito populations are highly variable. So we need spatial data. The first thing we want to know is what metrics are we going to use and how do we measure them? So what I've got up here is a basic primer to 3 metrics that I'm going to talk about today. There are more, Well, one is the prevalence of malaria. I'm going to call that X. What fraction of people are found with parasites in their blood? The second metric is called the entomological inoculation rate, or EIR. It's the number of infective bites that a person would receive per day. So 3 infective bites means I get bitten by 3 mosquitoes with parasites in their salivary glands. That seems like a lot, but that's actually on the axis of the data I'm going to show you on the right. And the last is R naught, the number of number of people infected per person infected. So it turns out that these 3 metrics can be transformed into each other under a model in a fairly straightforward way. And to get it, you have to realize that all the insect parts of R naught are related to this 4th metric I'm not going to discuss very much after this, called vectorial capacity. So if you think of entomological inoculation rate as being all the bites received by, the vectorial capacity is actually the flip of that. It's the number emerging from. So as long as all the bites land somewhere in this population, infected bites land somewhere, they're very closely related. So they're related by this formula. Vectorial capacity is the product of EIR and some function of X. And the function of X is talking about how infectious people are as a function of how much you find parasites in the blood. And when you look at these things, they're papers you're going to see cited at the bottom. They're sort of providing the technical materials. In the top right, you see a graph with a bunch of points on it. That's about 121 points taken from the literature where people had simultaneously measured the EIR and the PR. And you see it's a log scale on the x-axis. So there's a lot of variability here. But there is a statistical relationship between those things that we can characterize. And when we use that, we can actually make transformations from EIR to PR or back. And we can also make a translation then from EIR. And the part I can't see on my own screen at the top is a formula that relates R naught to vectorial capacity, which is related to EIR. So I can use EIR to estimate R naught. All right, so knowing that, we had to make a decision to what to measure. And the thing that we decided to measure was the prevalence of infection because it's measured a lot. We get a lot of information. It may not be the best, most informative metric, but it's been measured millions of times, whereas EIR has only been measured hundreds of times. Okay, so we get prevalence because we have so much more of it. So this is as of 2010. The amount of data has increased a lot. You can see where we had data describing malaria in the world. And you'll notice there are some very large gaps. DRC doesn't look very populated by points, for example. We assembled these, when I say we, starting at about the early noughties, I was part of the Malaria Atlas project, where we tried to gather as much of the data as we could find describing malaria, put it in a database. Later, that became the repository for open access data for the Malaria Atlas project. And there's a link at the top. And if you follow that, you can actually go download the data I'm going to talk about. Second step is we translate this into a map of malaria prevalence, which comes with an estimate of uncertainty. Now, this is actually projecting malaria prevalence in older children. There's a strong age pattern. So we normalize it to 2 to 10-year-olds. And what you're seeing here is a putative map of malaria in 2010. What you're not seeing here is underneath it at each pixel, there's a full representation of the full Bayesian posterior probability distribution function describing malaria. Now, it's boring to talk about uncertainty, so I'm not going to, but it's there. I promise you, when we use it when we do our analyses. The second thing is for each one of those times that we created a PR estimate, we then translate prevalence into EIR with uncertainty, and we get an estimate of the EIR. Now, as you imagine, since there's uncertainty in PR and uncertainty in the EIR, this comes with even more uncertainty. But we characterize that fully. And similarly, we translate that into a map using this relationship at the top into a map of R naught. So this is, in many ways, a global first. This is the first time anyone had produced a global map of the basic reproductive number for malaria, or for any disease for that matter. And so this was about 2010. How are we doing on time? I think I need to sort of, okay. Yeah, I'll try to get through this without feeling like I'm rushing. So now to use this, we need to actually have a measure, right? How much have I altered transmission and altered the conditions of malaria by doing things like giving people nets or treating malaria with drugs? And to do this, I have a different measure. It's called an effect size. So I'm going to walk you through a model we developed for insecticide-treated nets, not in a lot of detail, but enough that you get the gist. So we're going to start with a view of what happens when a mosquito tries to blood feed on a human. So there's a parameter here that phi, which is the fraction of times that that mosquito encounters a net instead of finding an unprotected human or a cow. What happens if it finds a net is it is either killed or repelled, in which case it has to try again, or in some cases, the net has a hole in it, or the person was out of the net, not using it, and so it successfully feeds, right? It takes the blood meal anyway. So what we do is we follow this through and we compute three quantities. What was the rate at which they blood fed at baseline? What fraction of the bites were on humans? And what was the death rate of the mosquitoes, or how long did they live? And then after we push it through this model, we do the same thing and produce those same three quantities, but now in the presence of nets at some fixed coverage level, we get a different measure of blood feeding rates, human portion, and death rates. And from this, then, we can produce an estimate of the effect size, which we're going to call a ratio. It's a ratio of two things. And since we've altered the conditions describing transmission with control, we invent something else called the adjusted reproductive number, RC. For those of you in the room, you can't see it because my picture is covering it up. But RC is like R naught, except for it's been modified by control. And the computation that does the effect size is the ratio of the baseline R naught to the modified R naught, the adjusted R naught. And it's just got those parameters, those modified parameters and ratios, so that the top has got the bigger number, if it's gone down. So an effect size of 10 would be a 10-fold reduction in transmission. Now I'm talking about an effect size on transmission, and when we look at this, we can talk about effect sizes on other things as well. But mostly we're going to focus here on effect sizes on transmission. And one particular example that comes up a lot, so I'm going to talk about it here, is the issue of global warming and global change in general and its effect on malaria. So what I'm showing you at the top right is a map that was produced in about the late 1960s by Lyshenko, and it was a guess at the distribution of malaria across the world using the best methods they had at the time. What I'm showing you at the bottom is our map converted to R naught. Or sorry, R map and PR. And then what you're seeing here on the left is a proportional reduction in RC. Now the message of this is pretty simple. Over a century that we saw about a degree or two of warming, we saw malaria's range contract substantially towards the equator. Now that was in a period while where the world was warming up. We saw a contraction in the range of malaria. Now this is not, for those of you who want to find a cause to trumpet for global warming, I'm going to tell you right now, malaria is not your cause, okay? Look for dengue or something else, but malaria is not your cause. The reason why, though, is not that temperature isn't making things worse. It's that other things are making things better by such a much larger amount that we don't see the effects of temperature. So things like economic development, things like vector control can produce, or changes in rainfall patterns can produce much larger changes in the distribution of malaria and malaria transmission than the comparatively tiny changes that are brought about by global warming. So it's not to say that temperature may not become an issue at some times and places. It's merely to say that it's not a dominant issue, that these other things, and we can use effect size to characterize the magnitude of the change likely to be caused by those things from various causes, and add them up in the same metric and in the same way. And this is what makes this metric in particular so useful. So if we want to do anything to translate those patterns about the past into the future, we need something that's capable of doing that. So we use simulation-based analytics. Now, what I haven't shown you is that the translations I've just done, we can do with models too. so that now we've fitted a model to the whole history of the past, and we're going to take these models and fit them to the future. And we're not taking the Ross McDonald model, we're taking this complicated set in a much bigger toolbox. So it's not just a crescent wrench. I mean, we got hammers and we got saws, we got everything. And I'm going to walk you, if you imagine this is like Sleepless in Seattle, this is the montage part where I'm going to walk very quickly through a lot of the work we've done, which has occupied most of my time over the past 20 years. And just give you highlights of things we've done. If there's any particular one you're interested in, let's talk about it. But we've reframed models for malaria spatial dynamics in at least 2 different ways. So we have a micro and a macro. So micro deals with very small areas and the distribution of resources in bodies of water and places where mosquitoes blood feed. And on the right-hand side, we deal with a more traditional patch-based metapopulation model. We've reformulated all of the models so that they're spatial and we can apply them to places with actual parameters. We've reframed the models of malaria epidemiology. Those of you who are at home can't see that I'm pointing at my graduate student, John Henry, who's been working on this. And what we've managed to do is to take the very complicated process of malaria epidemiology and simplify its representation in a way so that we can make hypotheses about the effects of control on things like disease and prevalence. And so we've added a few equations, but not too many. About 3 equations give us most of what we want to know. We've reframed the models for mosquito survival and movement. We've reframed the whole notion of the human human blood feeding and the human biting rate to include things like heterogeneous exposure and travel, so that and the fact that mosquitoes are in a particular situation where there may not be a host that it wants available. We've modified the models of infection dynamics so we can analyze, for example, infection dynamics with noise, because weather creates a very noisy pattern. We've reframed the models of mosquito population dynamics, in particular linked them up so that we can look at population dynamic feedbacks from adult vector control. We've built models of travel. And so on the right-hand side, you're seeing analysis we did of malaria connectivity in Kenya based on Safaricom phone records. On the left-hand side is a very simple model we did of malaria importation into Zanzibar, where we look at all the pathways that can bring malaria into a place that's trying to eliminate malaria. And we've built model after model of vector control so that we can look at things like larval source management, insecticide-treated nets, indoor residual spraying, and their effects, especially in cases where they cause mosquitoes to move around rather than to be killed. So this gives us this very large toolbox, and the way we do our analysis is we pick a model from the, pick a tool out of the box for each thing, put them all together to create a model and do an analysis all the way from beginning to end. That analysis looks like this. Take the data, gather it all for a place, recreate the history of the past. So what happened? What do we think actually happened here? And that comes with a lot of uncertainty. Next, we try to reconstruct what would have happened had we not had any control measures in place. Third, we sit down with a program and we ask them what they're trying to do under their budget. What else would they like to do? And we do scenario planning. We evaluate those scenarios with the simulation-based analytics, and we find out which of these are most likely to save lives for the money. Now, remember, we've done this many times with many different models. So at the end, we come up with a policy ensemble. Each one of these analyses, since we've automated it, gives us a different bit of advice. We sort through that advice to find out what recommendations are likely to be supported. Now, if every scenario came up with the same answer, we have our answer. There's a consensus and it's robust. If there's a difference in some of those scenarios by analyzing those ensembles, then we have to go back and figure out what was the difference. And then our advice comes with a most likely alternative and an advice about what to measure. And this puts us in a position now to finally do adaptive malaria control, where we can sit down with a program in an iterative way, and year after year, replicate the analysis, trace what's changed. So when we change something in a model or we change an assumption or we get new data, we can track it back and tell them why the advice has been changing. I'm just going to give you one example and then I'll some conclusions. So this is a picture of malaria on Bioko Island and some analysis that we did. So one of the problems with oil economies is that people are coming in and out of them quite frequently. And this oil economy is no different. So what you're not seeing is population density, which is concentrated in the city in the north of Malabo. So about 80% of the people live in a very tiny area in the north, and it's very rural around the outside. But people who live in Malabo tend to leave Malabo and go off island. People who don't live in Malabo tend to leave and visit Malabo. And then we found this through asking people, about their travel patterns and travel habits. When we put these travel patterns into a model, something jumped out at us. It looked like in Malabo, almost all of the malaria that you saw could be explained by travel off island. People going somewhere out of the island and coming back with doing that enough times that it would account for all of the malaria you've seen. Whereas around the outside, it looks like there was still ongoing transmission. So we tried to do an analysis to help them decide where to put their vector control measures that took this into account. And so this is what you're seeing is in the top, you're seeing our estimate of the travel fraction. And the more yellow it is, the greater the fraction we thought came from off island. And what you're seeing in the bottom is an estimate of our uncertainty. So the darker it is, the more certain we are. And similarly, if you took that away, this is what we predict would be the residual fraction of transmission that, what would be left is at the top, and you can see the estimate of uncertainty is around the bottom. So this is being used now to try and send a couple of messages to the program. One, you have a lot of malaria being imported onto your island. You're trying to eliminate malaria, but it's going to be very difficult to eliminate a disease that's coming in on an airplane. So you're going to have to either do something to control malaria in travelers or give up on the idea of malaria elimination. It's not something they want to hear, but it's a tough message. And the second one is that there are probably places where there's ongoing transmission despite all their best efforts. And those are the places where they should focus their control. All right, so I'm going to go to what did we learn? And then wrap up. So one of the lessons learned is, it's going to sound really silly. A good approximating model shouldn't leave out anything that's really important. Okay? I know that sounds silly, but a lot of the models that we were playing with initially left out malaria importation. And it ended up really making it hard to give good advice because we couldn't get our models to fit. And the only time that we could get them to fit is when we put that pattern in, and all of a sudden then it just kind of popped on the page. And we were like, oh my gosh, that's what's going on, right? So don't leave out anything important. How do you know what's important? You figure it out as you go. Models should be realistic enough to interact with the domain experts. So if you're talking about drugs, you want it to be realistic enough that when you talk to somebody who works on the drugs, they can follow what you're doing. Or if you're talking to a programmatic person, it has to have space in it so you can see where they're recommending to do things. And if you're talking about vectors, you need to talk in the language of the vector biologists. So that's part of what we learned. Another lesson is that spatial complexity has always got to be there because the spatial variability is always important. But secondarily, and this bit us in the butt a couple of times, you have to put exogenous forcing in the models. Okay, what do I mean? You make a projection and you leave the weather out, and next year you have a bad rainy year, you're going to get in big trouble with your funders. Not going to go into detail, but you can imagine if you promised it was going to get better and it got worse, and then you're standing there saying, well, yeah, but the rain, you look kind of stupid. If you told them in advance, things are supposed to get better, but if we have a rainy year, there's nothing we can do about it, then you've covered your butt. And part of this is, satisfying your funders. I'm going to kind of skip a lot of lessons and just say this. When we got involved in this process, I was trained in ecology and evolutionary biology as a scientist. And I'm convinced that this business of mimesis has sort of been taught badly to me and many other people like me, because what makes a model good depends on what you want to do with a model, right? There's not always one best model. And so this business of trying to find the one true way to represent a model is always bad. And if you guys will remember that scene when Neo wakes up from his nap and the computer's talking to him, it says, "Follow the white rabbit." He goes to the door, there's a girl with a tattoo, a white rabbit tattoo. He's holding a book called Simulacra and Simulation. It's a book on literary theory by a guy named Baudrillard about mimesis. These guys who are building the matrix and that sort of whole game of representation knew what they were doing when that book in his hand, right? The theme of simulacra and simulation, it's very, so without doing it, is that sometimes you can get further and further away from the truth by trying to represent various degrees of separation from it. And so theory plays an important role in science, but it may not be the best thing because it distorts things, right? It's a distortionary thing by definition, and it's trying to abstract these principles. It may not be the best thing to represent the truth to the people you know. So you can always give generic advice with theory, and it's good to start simple, but you probably don't want to stay there forever. Oh dear. Last slide. Oh golly, I'm going to show you that one. If hyperrealism comes up, I'll give you that. I just want to give some acknowledgments and thanks to people, funders, Bill and Melinda Gates Foundation, NIAID, NSF, BMAP Rapid. We just got funded to work on some of these same things for West Nile virus with the Microsoft research. Mike Reddy is here representing that team, so we're very excited to start doing some of the same things for West Nile virus with Houston, Harris County, Texas. Second line are people from my lab here at IHME and other places, including John Henry, who's in the audience, Austin Carter, my current students, and Sean Wu, Hector Sanchez. The whole MAP team, Simon Hay founded it. It's now run by Pete Gething. You were asking me about Andy Tatum. Andy Tatum is in that crew. There's a team I've worked with for over sort of more than a decade of research in Uganda with UCSF. Moses Kamya and his team in Kampala at IDRC. Guillermo Garcia, Charlie War, Carlos Guerra, and the BMEP team who we work with in Equatorial Guinea. Sam Kawari, Fred Akumu, and the IHI team. Malaria Modeling Consortium, which I didn't bring up, but it's another way to get a robust advice is to have lots of other people do it in the same way. And then a lot of friends who have helped me along the way, and Jimmy Opago, Damien Rutazana, and the Ugandan Ministry of Health. So thanks, I need to mention all those guys, and then I'll take questions if there are any. Thanks. Are there any questions? Yeah.
That's the question, but remind you to repeat the question for people who are on.
Okay.
Talk about elaborating models. I think you did a nice job sort of talking about how you elaborate models that don't do too much. You're trying to find the right model and the right combination of models. My question has to do with your point about your process, which involves fitting models and then applying models. You ever you have a process by which you evaluate the risk of overfitting? As you elaborate models, as you include more processes, you talked about agent-based models, which have lots of processes. Some of the things you're talking about were elaborate processes with lots of parameters that then if you have data, you have you set the data to the parameters you don't fit, it result in models that are too fit to the data that you have. And what you're trying to do is that what happens in applications where we don't have data.
So I think this gets at the heart. So the question is about so how do we do model fitting and how do we avoid overfitting? Because these are complex processes. We have complex models that we're fitting to the processes. So this is what I'm going to go back to the bit I said about master policy and trying to remember what it is we're doing. The other thing is, are we doing something to analyze the past or make projections in the future? Right? So the past and the future are very different things. The past, you can't change it. There may be data that you didn't find, but you can't collect any more data in the past. And the future, you don't know anything about the future unless it's like the past, but you can collect data in the future. So those are the big differences, right? So when we do statistical analysis for science, we worry a lot about overfitting. And we do ROC curves and we sort of out of sample validation. When we try to understand the basic relationships driving the patterns in the data, we as scientists, we worry a lot about overfitting and we try to pick the simplest models. The problem that I worry about a lot is, especially with the epidemiology side, there's a lot of identifiability problems where the same mechanism, or different mechanisms would produce the same patterns, and you only have a pattern. You can't conduct the experiments for ethical reasons or other reasons. And so you get a range of models that could fit the data equally well. So when we turn those around and now become analysts, we throw parsimony out the window. And we do it on purpose because we want to deliberately characterize the uncertainty and span the uncertainty. So all of those mechanisms that could be at work when we tried to fit, we now turn them and say, well, let's give them a head, right? And then there's another part of the problem, which is, I guess the best way to explain this is, for me, I was sitting, it was a very nice vacation. It was a work vacation in Orvieto, but they gave me some data and they said, we need an algorithm to do age correction for prevalence data. Here are 210 pairs of points, fix it. And I was going out of my mind trying to do it until they came back and they said, oh, we found more. Here's another 500 points. Then all of a sudden, patterns started popping out. And I was like, oh, I can do this, right? So part of the problem is when you do an analysis with a partial data set, you're going to get an answer that's going to be very different. And if you just focus on the fact that you're trying to do the most parsimonious answer and simulate based on it, you probably dramatically underestimated the uncertainty. So when we do science, yes, we're very concerned about overfitting, but I'm not so sure that when we try to be robust and propagate all that uncertainty, that overfitting is such a bad thing, especially if you're creative about overfitting. Because when you start projecting those into the future, you're generating hypotheses that in some ways go beyond the data and that might actually be true. And the only time that they would matter is if they're going to affect a policy. You see, we're not trying to say something about the process anymore. We're trying to make a decision about the policy. And so if uncertainty that could be, called overfitting is affecting the policy, well, when we go back to scrutinize it, then we worry about it. Well, maybe it's just, we're fitting the noise. But I do think that it calls for different criteria for determining whether you're going to overfit or underfit when you're sort of talking about how to propagate that uncertainty forward to make a decision. Long answer. Mike. Thanks. We're still sort of working on that, but mostly it's coming in from the mainland, Equatorial Guinea. Probably both ferries and flights, because now I think the costs, you probably know more about this, the cost of flights has come way down, so it's about the same as the cost of a ferry now. And so you get people from the same SES, socioeconomic status, coming in. The one risk factor we did identify was being a government worker. Was a really, strongly associated with being, having a history of travel and being infected. And that's partly because they make a requirement that all of their government officials spend a, large amount of time on the mainland. So they're going to, occupy, or, cities where there's still highly endemic transmission on the thing. The other thing that I think is really interesting about it is we did discover that people who say they've traveled a lot tend to live next to people who say they've traveled a lot. So this is not really that surprising if you think about it as a demographic problem. But if you think about it as a statistical problem and you just assume everybody's randomly distributed, then you'd get one answer. So the fact that people who are infected and live next to each other have all traveled suggests that it may not be local transmission. It may be that they have a shared trait rather than that they have a shared transmission risk locally. So that was in one.
Thanks, Tom.
Thanks.
Just sort of piggybacking on that, you just commented about how commonality of being some of the travel that's supposed to be a more efficient. Does travel in these areas fall along associated factors, strata and see?
Yeah, I'm going to remember to repeat the question this time. So the question is does habitual travel follow along socioeconomic lines? And I think undoubtedly the answer is yes. So people are there for the oil jobs are traveling a lot more. Some of them didn't bring their families, they're living temporarily and going back and forth. And this is also partly explaining the pattern we see on the rest of the island, where most of the rest of the island is made up of native Equatorial Guineans, Equato-Guineans. And they have never had a presence on the mainland. So they're going into Malabo for, to go to hospitals, to go to government bureaus, to get restaurants or conduct business, going home. Whereas the people, the other people. So government workers, one identifiable class, oil workers, another, and native Equato-Guineans are sort of clearly all three identifiable groups. There's also age. So what we're trying to do statistically is sort it out and try to model it. So, you do all your statistical analysis and you turn it around and you say, okay, let's suppose that we use these covariates to predict frequency of travel with some uncertainty, then we can make a guess at how much we're going to affect transmission. And so that's where we are at the present time, and sort of sorting through some of those kinds of answers to try to figure out what's going on. All right, thank you so much for having me. It's been a great pleasure. Aside from the technical difficulties, victory, right?
Yeah, well done. Thank you.